There I am. So for those of you who don't know me, I'm Chantal Stern. I'm the director of the Cognitive Neuroimaging Center here in the Kilachon building. And I'm a cognitive neuroscientist, and I have studied the brain for many years now, generally thinking of it in terms of its neuroanatomy and also its function in terms of behavior and cognition. And more recently, what I learned from my students who primarily had been taking a network statistics course with Eric Kolachik was to start thinking about the human brain as a network. And so this is completely new to me, and I'm going to just introduce a few of those terms to you as they may be new to you as well. And then I'm going to give you a couple examples from papers that have been done in my lab recently using these methods. So first of all, what we can do is define a node. So a node is just basically a brain region of interest, so shown here. We can also define edges, which is basically just functional connectivity or a correlation between two nodes. And for us, we're using this as the bold signal to create these edges. And we can also develop communities. Why is this not? It's delayed. Communities, which is basically a group of nodes, which can be defined in sort of an a priori manner or a data-driven manner. So I'm going to show you some of that in the next slide. For our lab, we generally have been using this parcellation, the Schaefer parcellation. So it divides the brain into 400 different locations, 200 in each hemisphere. That's actually what we use as our nodes. And then we can actually look for the connectivity. So on the right-hand side here, this is a paper spearheaded by Kylie Eisenberg in my lab, along with a collaboration with David Summers' lab. And on the right-hand side, you see this node-to-node connectivity when participants are doing a memory-guided attention task. So what do I mean by that? If I asked you all to go outside and find the sit-go sign, you could all do that because you could localize the location based on your long-term memory representations. So what we see here on this next slide is basically how these networks reconfigure to support memory-guided attention. So you see the networks that are all shown on the left-hand side, these different colored networks from the Yao Atlas. And you also see these very specific node components. So on the top, you actually see increased connectivity, and on the bottom, you see decreased connectivity. The size of the blobs is basically showing you the increases in activities, the size of it for these specific nodes. Particularly, you can focus on these midline, these posterior midline areas that include the precuneus, the retrosplenial cortex, that are facilitating these components of long-term memory-guided attention. And just to jump very quickly to another study that was done in my lab, primarily by Tom Marin, and also in collaboration with Joe McGuire, uses these dynamical network analysis methods to look at changes that occur across time. So again, in this case, we take these 400 nodes from the atlas, from the Schaefer atlas. And in this case, what we're doing is we're looking at the networks across all 9 runs. So the subject is learning a rule, or actually a whole set of rules, across a long period of time. And we can look at network changes across time in individual subjects. And what we find is that participants who are good at learning the rules, as opposed to those who are not successful, actually develop a very stable network, sort of brain network architecture. So if you're interested in that, in this sort of analysis, Danny Bassett will be speaking on Thursday as part of the Hariri Center, and this is work that she developed And I'll just end there by thanking both Boston University, the Kilachon Fund, as well as NSF and ONR for funding the work. Thanks.